Hi everyone. We'll just wait for maybe 2-3 minutes and then we'll get started. I hope everyone can see the screen. Okay, then I think let's get started for today. Um, so today's problem solving session and discussion session will be about the coursework that you guys have received for week 6 of your course, which mostly involves chromatin organization in the nucleus and also the importance of histones, as well as a lot of detail on some non-histone proteins that are important for chromatin compaction and condensation, such as the SMC, structural maintenance of chromosomes, right? And you also had some guest lectures from various professors going into the details of how these loop extrusion experiments were um, conducted, which were very interesting, right? The real-time imaging data that Dr. Mahipal Ganji showed, as well as the story of essentially how SMC proteins were discovered, as well as the various interesting aspects that come up when these came up when these experiments were carried out, and how the hypotheses had to be modified based on the many chip assays that they did. So with that, let's get started. I'm just going to go through a bunch of different topics that I thought were kind of useful to go into detail about, and then we can look at all the questions. Okay. And for those who don't know, when we go to the questions, I'll read out the question and then you guys can answer by unmuting, or if you're more comfortable with texting it on the in the chat of this meeting, then you can do that as well. Right. So starting off right away with some genome facts. So as you guys know, compaction of the genome is very important, as you must have realized that if you have a genome size of 3300 megabases and you have 25,000 genes being expressed by it, this is going to occupy a lot of space, right? 2 meters or so. But we somehow managed to fit that into our nuclei, which are so tiny. Like a cell is what, a couple, like 10-20 microns. Within that, the nucleus is 5-10 microns, max to max. So within that, we have to compact all of our DNA into these chromosomes, and they are going to be there. So I just thought it would be, I mean, interesting to see just how much, how huge the genome size can, how much it can vary between organisms. It doesn't seem to matter. So there was this like a hypothesis where they essentially uh, it was like something that confused researchers a lot, which is that with the size of the organism increasing, why isn't it always the case that there are more genes, right? So the C value paradox or something it was called. And this is so directly opposite to that, right? Drosophila has 165 MB genome size. Um, and like C elegans has 97. So uh, yeah, anyhow, I thought that might be interesting. And this is a karyogram, or yeah, which basically shows the banding patterns of a lot of chromosomes. So this is for humans. You can see 22 different chromosomes and how they have sort of delineated the banding patterns of these. And we'll go into what banding patterns are in a little while. So for all of the chromosomes, this you would typically see this kind of structure in a metaphase chromosome when they've condensed and doubled up. And of course, they've also interestingly shown some mitochondrial DNA, because that's another important part that you inherit from the oocyte uh, during fertilization. So each person inherits mother's mitochondrial DNA. And you have also then the X and Y chromosomes here as well, uh, shown here in the karyogram. So compacting this DNA into something that will fit inside a nucleus obviously needs some very important factors that can help do so. And one of these factors is histones, right? And all of us have heard about histones by now, I'm sure. So here, this is just a demonstration of how DNA is initially compacting into these little nucleosomes. Each one will have about 10 nanometers or 11 nanometers diameter, and they form this bead on a string structure, right? And there are, I think, 147 base pairs in between each of these beads. sorry, no, I mean, there's about 80 to 200 nucleotides between these beads and 147 nucleotides in one turn around a nucleosome. And so these beads on a string are then even further compacted into this 30 nanometer fiber, which is then compacted further and further and further and further until we have something that looks much like this chromosome, which is a metaphase chromosome. And this and its replicates all manage to fit inside the cell. So maybe it's important then for us to take a closer look at histones. So as you have studied in your previous studies as well as in this coursework, right? Histones are very important proteins. They're basic proteins. They're highly enriched in lysine and arginine, which makes them quite positively charged on their surface. And they are between 100 to 130 or so amino acids in size. Um, and typically with DNA, it forms an octamer. So this structure, you will have a tetramer and a tetramer, and these two will bind together along with the DNA looping around it, and that will form a histone octamer. And the positioning of histones and modification of histones can actually also determine a lot about gene expression in these regions. So a lot of studies about epigenetics deals with the modifications and different alterations of histones. So you can have open chromatin or euchromatin, which is typically more loosely packed. So it's more um, accessible to transcription factors. So that makes transcription easier to occur over there. And you can also have closed chromatin or heterochromatin, which is more densely packed, and transcription cannot occur that well in those regions. So those genes are essentially transcriptionally repressed. And histones play a part, as well as other proteins, they play a part in these epigenetic modifications, which determine whether a gene is being um, transcribed or not. So whether that, and if it's been transcribed, you're going to produce an mRNA. And if you have an mRNA, then typically you would also then produce some protein with respect to that mRNA. So histone modifications can overall affect the functional readout of a cell, because if you don't have some protein or you have more of a protein, obviously that is going to affect how well the how the cell utilizes those proteins and how it functions, right? So as you have seen in your coursework, histones come together to form an octamer. So typically how this happens is that you have these different subunits. So you have H2A, H2B, H3, and H4. So H2A and H2B come together and form a heterodimer, and H3 and H4 come together and form another heterodimer. And then you have a duplexing of this dimer. So you have a tetramer of H3 and H4 and a tetramer of this. And these two come together and form a histone octamer. So that's how you have this structure forming. And then you have this DNA loop around it, which will interact with these histones, approximately 147 base pairs. And that entire structure will be called a nucleosome. And it's useful to know that all of these histone proteins actually have a common structural motif that was mentioned, right? Called the histone fold, which has a couple of alpha helices, I think 3 alpha helices and 2 loops. And they have an N terminal that's quite flexible and amenable to a lot of post-translational modifications, which will be important about when it comes to the regulation of gene expression, loosening or compacting of DNA, right? So yeah, so that's the structure of the octamer with the DNA. And of course, how come the DNA is staying there? It must be interacting with the histones, right? So there are a lot of different interactions that take place between histones and DNA, from hydrogen bonds to non-polar interactions, salt bridges, um, charged interactions, all of these things. I think there's like 4 or 5 different types of interactions that are taking place between DNA and histones that make sure that the DNA stays in its place. Along with that, of course, there are also non-histone proteins that are important in compacting and attaching the DNA in different ways. So I thought I'd go a little bit more into detail into H1 histone. So here you'll see that there is no mention of H1, but H1 is another histone that's not part of this initial nucleosomal core particle, but it's very important for making sure that the loop of DNA around in the nucleosome stays well fixed in its position. So this is H1. It is a linker histone 1. Okay. And I mean, there are multiple H1s based on whatever organism it is. And it's a very important component of chromatin because it binds to the nucleosomal core particle around where the DNA enters and exits. So entry and exit meaning where it initially enters before looping and then when it exits. So at those sites, it is binding there and it will stabilize both the nucleosome structure uh, with respect to making sure that this DNA loop remains tightly wound around the histones and also with respect to higher order architectures, which we'll see in some time. So it seems that in general, H1 molecules consist of a central globular domain with a more flexible tail region, which has N and C terminal ends. And typically, if you have a flexible end, they may be actually amenable to different modifications. And these modifications can obviously alter the way how tightly the DNA is bound, how well it is holding on to it, and different kinds of and change the transcriptional signature of that region of the genome as well. So H1 histone modifications can also alter how different genes are expressed. And there is an interesting paper that I have cited somewhere along in this presentation that has actually, where the authors have actually looked into mapping um, H1 across the genome. So where all H1 is there in the genome. They've done some extensive chip assays and other assays to look into that. And so then that, if you know where H1 is binding, you'll have a better idea, or you'll start to have a better understanding that if we were to say knock down or knock out H1 histone, how would these genes then be affected, right? So this is from a different paper where they were essentially looking at whether histone 1 is altogether needed in the cell or not. So it turns out that in eukaryotic cell, um, if you were, I think if you were to do a knockout of H1, which is what some people had done in tetrahymena cells, they had depleted H1 and they saw that it was not completely essential for the organism and it actually survived. And this was quite surprising, even though obviously there will be some subsets of genes that are downregulated or their expression level has changed. Overall, the organism was fine, it was healthy. And that was, quite surprising that it's not, because other histones, if you knock them out, the organism is actually, the organism dies. So it's typically embryonic lethal. But H1, in the case of H1, knockout of it, if it has multiple subtypes, so it's not easy to knock all of it out, but a substantially lower amount of H1 does not lead to embryonic lethality. So, yeah, so here in this paper, they have done a knockdown, and this is just this is like a fly, and they've shown sort of how the external morphology of the fly has changed. This is the control, this is the H1 knockout over here, and this is the lower region of the fly, and this is the head of the fly. And you can see there's some changes in the color and, some changes in this. I'm not really a fly expert, but whatever this stuff is near its eyes, it's maybe some feelers or something. It's the change has come in the morphology over there. So, and this I found an interesting paper. Okay, so this is a continuation of the same one, this paper. I think I can point. Yeah. So what they did is this H1 knockdown, right? So it's a depletion. So where in this Western, they've basically shown that H1 has gone down, and also these are some heterochromatin related proteins, HP1, and this repressive marker protein, H3K9ME23. So these are some proteins that are found prominently in these bands and interbands, and they can give you a banding pattern. So here you'll see in the control, you see that this is the DAPI stained one, this is HP1. In the knockdown, you'll see that the way HP1 is localized has changed. In B, oh, and I have to mention that these are the polytene chromosomes that are found in the salivary glands of Drosophila and some other insects as well. So typically, they have these banding patterns, so bands and interbands of these polytene chromosomes, and they have some prominent chromocenter staining where there will be HP1 and this H3K9 protein. And what they saw is that depleting H1 resulted in a lot of altered chromosome structure, and there was a loss of this banding. And H3K9ME2 staining was completely gone. So that would indicate that these chromocenter points were disrupted. And also HP1, just the patterning of it had changed. Like the way the foci sort of came up is very different from here. And you can see, I think in the next one it will be more, it'll be easier to tell. So I'll just go to that. Yeah. Here you can see that this, again, this is another polytene chromosome that they have stained. And this is basically some gene that you can mark out with the help of a protein. So you can't bind an antibody directly to that gene. So what you can do is there is another um, sort of site which you can use an antibody against because it's expressing ectopically. And what you can see is in this control, you can see how the banding pattern has been sort of elucidated over here in B. Whereas in the knockdown, you'll see that that banding pattern where you can see this protein, this GFP LAC-I, you can see that the banding pattern has completely changed and even abolished in certain regions, right? So that is showing essentially how the gene expression is changing, and if the banding pattern is changing, that would definitely indicate that the distribution of euchromatin and heterochromatin is also changing in an H1 knockdown condition. So I thought this might be interesting for you guys to sort of see the real life effect of removing one histone. Of course, this is not easy to do it if it were to be anything other than H1, because the other ones you'd have to maybe get like embryo cells or something. But in this case, at least the organism survives, albeit it has some gene expression changes. Right. So another set of proteins that are very important for sort of transcriptional control, repression, or activation of different genes in the genome are these HATs and HDACs. So HATs are histone acetyl transferases and HDACs are histone deacetylases. And these function antagonistically to one another and they control histone acetylation states that are very important for many cellular processes. So acetylation is generally associated with transcriptional activation, and there are several HATs identified. And similarly, the activity of HDACs, which is histone deacetylation, is generally associated with transcriptional repression. So how does this happen? I think I have an image for it. Huh. So essentially, you have HATs transferring an acetyl group onto a lysine residue, an amino acid lysine, right? And um, this is a negatively charged group. Okay. So now if you have the transfer of a negatively charged group to a generally basic protein, so if you have acetylation of many regions of the histones, what would happen? You would neutralize that that positive charge that's found typically on the histones, and it would overall either become neutral or positive, right? But now the histones being positively charged, that actually helps them interact with the DNA, correct? Because DNA in the phosphodiester backbone actually has a lot of negatively charged groups, right? The phosphate groups and so on. So the positive basic residues of the histones interact with the negatively charged DNA backbone, and that helps this structure remain a lot more closely packed. But the minute you have a hat that's transferring this negative group and neutralizing the positivity of these histones, you're going to have essentially loosening of the DNA. And if the DNA gets loose, what will happen? You will have regions loose from the histones, as in, so if it's less tightly packed or it's more loosely packed, you will have regions that are more accessible to transcription factors. And so you can have transcriptional co-activation of some genes if there is hat activity in the histones that is near where that gene is located in the genome. And the opposite is true for edge tags. If edge tags begin to remove a lot of these acetyl groups that may be there um, sort of constitutionally in some regions, and then edge tags will act to um, remove those acetyl groups, you're actually increasing the positivity in that area and making the DNA more strongly attracted to that histone region, and it'll be tighter packing and it'll be harder for the transcription factors to access that DNA. So that can lead to transcriptional repression. And so these enzymes, these H hats and H tags, these are actually quite highly conserved from yeast to human, and based on their homology with yeast orthologs and other analyses that people have carried out, there are actually 18 edge tags in humans, and they can be grouped into class 1, class 2, class 3, and class 4, where class 1 has edge tag 1, 2, 3, and 8, and that is homologous to RBT3 in yeast. Class 2 has edge tag 4, 5, 6, 7, 9, and 10, which is homologous to HDA1. And then the ones that you guys might have heard of more from previous studies with this class 3, where you have these cert 1, 2, 3, 4, 5, 6, 7, and these are homologous to cert 2. So these are quite well known. I think a lot of people have heard of them, and people are targeting these certs a lot with respect to um, metabolic rewiring and anti-aging function and stuff. And then you have the class 4 edge tag 11. So similar to hats and hats and edge tags essentially have a lot of very important functions in cellular pathways, and if they are misregulated, obviously what can happen is that you can have, say, one gene expressing too much because you have excessive hat activation in that region, and that's just making all the DNA in that region loose. It's making it accessible to transcription factors, and then maybe this oncogene is just highly expressed because of some random hat mutation. The other way around could also be the case, where you have an edge tag that is mutated, and so it's not properly condensing the chromosomes, and that's leading to transcriptional overactivation. Or the other way around, where you have some tumor suppressor gene that's unnecessarily being repressed because of some overly active edge tag. So either way, if there is a mutation or some dysregulation of hat or edge tag, that can lead to a lot of cellular um, problems and also cancers. And in fact, there are quite a lot of therapies now that are coming out with different inhibitors, chemicals, antibodies, whatnot, that inhibit mutant activity or inhibit HDAC activity in general, that can be a part of anti-cancer therapies. And so I explained this. This is basically, a reversible reaction where you have an acetylation of this lysine residue, where a hat can transfer an acetyl group from an acetyl-CoA to the epsilon amino group of the lysine residue. And with the help of a water molecule, an HDAC actually can promote the removal of the acetyl group from acetyl lysine, and that will generate that free moiety over there, and it will release the acetate molecule. So these are the various things that people have been studying. As I mentioned, this kind of metabolic rewiring with resveratrol and calorie restriction, that's to modulate the activity of cert proteins. And yeah, there's these different inhibitors where you can inhibit HAT, you can inhibit HDACs, and also people are studying about the environmental and more sort of macro level changes that can affect HAT and HDAC activity. So epigenetic modifications with respect to physical activity, nervous stimuli, immune activity, and then even your diet and other sort of risk factors like stress or smoking. So they are of a lot of interest right now, and the field is, I'm pretty sure, quite booming with studies on HATs and HDACs and how we can leverage what we know and the molecules that we know about and use them for therapies. And this is sort of a perspective on how they can be used in terms of cancer therapies, as I mentioned earlier, where you have HATs and HDACs that are in the um, nucleus that are sort of changing around the accessibility of DNA. And you can use a HDAC inhibitor here that would loosen or make some regions more accessible or less condensed. You can also sort of use HATs to say promote p53 activation. So promote p53 acetylation. HSP90 is a heat shock protein. It's something that helps hold the DNA. And it's a chaperone protein. And that can be acetylated, which is important in, I think, the unfolded protein response. And you can also inhibit HDACs and improve acetylation of NF-kappa B, which is important for inflammation, or tubulin, which is important to make sure that the cell has proper localization of cytoskeletal elements like microtubules, and it can essentially give them better shape because softer cells in cancer typically lead to higher proclivity of metastasis. So it can um, cytoskeletal dysregulation in cancer cells is a major way in which cancer cells can get away. So if you were to acetylate tubulin or the genes for tubulin, you could or the tubulin protein itself, you can actually improve the way these structures form. Um, yeah, and this is sort of just showing that um, where you can modulate these different processes, modulate cell cycle, modulate apoptosis, try to increase apoptosis in cancer cells or um, yeah, using different therapeutic strategies against HATs and HDACs. Does anyone have any questions till now? I'll move on otherwise. No, all good? Okay. Right. So before going into sort of these higher order structures formed from, uh, just a second, just a second. Yeah, sorry about that. Okay. Yeah. So chromomere uh, is like a unit of these higher order structures. So I thought it would be useful to know what these chromomeres are before we go into studying about, say, polytene chromosomes or lamp brush chromosomes, which you guys have, I'm sure, heard of and maybe even isolated in your practicals in your colleges. So a chromomere, it's also sometimes called an idiomere. It's one of the serially aligned beads or granules of a eukaryotic chromosome. And this results from local coiling of continuous DNA threads. So what does that mean? It means that there are regions where the chromatin has been compacted a lot through some localized contraction. So we know about the compaction with the help of histones and SMC proteins. But there is some localized contraction as well that increases how compacted they are in a sort of in certain regions, um, based on uh, yeah, based on sort of the genes in the area. So how you want a heterochromatin versus euchromatin banding pattern. So in these areas of chromatin where there is no transcription, condensing of DNA and protein actually results in the formation of chromomeres. So where you have more heterochromatin, you have less accessibility of DNA, you will have more compaction, and you have the formation of these chromomeres. And they are actually organized in a discontinuous linear pattern along the condensed chromosome. So in you guys remember from your studies about meiosis that in prophase 1 of meiosis 1, you actually have these different phases, right? So you have pachytene. So in pachytene chromosomes, where you are aligning them up and preparing for, you have the loading of the synaptonemal complex and crossing over going on. During that time, you have these chromomeres organized in a linear pattern along the chromosomes. And the linear pattern of chromomeres is linked to the arrangement of genes along the chromosome. So it's, as I mentioned, it depends sort of upon what genes are where. These chromomeres and the condensing in those regions can come accordingly. So sometimes the genes, sometimes it has genes, sometimes it has a bunch of clusters of genes within the structure, and these aggregates of chromomeres can also be called as chromonemata. And these cohesive proteins, SMC3, and I think I meant to write cohesin, my bad, and RAD21, these actually play an important role within these chromomeres at higher concentrations. They help with maintaining the proper structure of chromomeres. So this is one example of a higher order structure where you can see these banding patterns, bands and interbands. And these units are called chromomeres. And you can see it sort of zoomed in over here. So now with that understanding, let's look into lamp brush chromosomes. So as you know, I'm sure you are generally aware that lamp brush chromosomes are a very special type of chromosome. You can see this here. This is a drawing um, taken from quite an old micrograph. I think 1905 someone had drawn this. So I found this on Wikipedia. So this lamp brush chromosome, it's found in the growing oocytes of most animals, except for mammals. Okay, interestingly enough, and they were first described by Walter Fleming and Ruckert in 1882. So the chromosomes, they transform into this lamp brush form during diplotin stage of meiotic prophase 1, and this is due to an active transcription of many genes. So always these kind of compacting and all of these condensing and all, they are not just random. They happen due to some specific tendencies of genes, whether they're transcribed highly or transcribed less. This is very complex process. A lot is not known about why or how it's determined where this banding is going to take place or where this compaction is going to take place. So in this case, it will happen because there's an active transcription of many genes in certain areas, and they are highly extended meiotic. So what happens here is that if you form this lamp brush um, during diplotin, they will have a highly extended meiotic half bivalence. You guys remember what a bivalent is, right? From when we studied about meiosis, right? So it's essentially, I don't have an image here, but it's essentially you have the homologous chromosomes that are attached and undergoing crossing over, right? So these are the half bivalents. So they will have two sister chromatids. And the lamp brush chromosomes are very clearly visible. Even in the light microscope, they're so like big and so condensed that they're quite easily visible. And you would think typically that chromosomes are more visible during metaphase, right? Where you have them sort of aligned, you have them fully condensed. Um, but in this case, they're just visible from this pretty much prophase one itself. And they're organized into a series of chromomeres with a large loops that extend laterally. And these loops are important because they are actually regions where there's a lot of transcription that's taking place. So as they've mentioned here, that continuous RNA transcription is required to maintain typical chromomere loop structure of these lamp brush chromosomes. So these loops that you can see here that I'm showing, these have a lot of transcriptional activity going on there. And if you inhibit the transcription, this leads to retraction of these lateral loops into the chromomeres and chromosome condensation takes place. So these lamp brush chromosomes produce a large number of mRNAs. If you have a high amount of transcriptional activity, you're going to have a lot of mRNAs and non-coding RNAs that are produced and synthesized from these lateral loops. And these actually seem to be important for oogenesis and the early stages of embryogenesis. So it's not just random. Again, there is a reason, and it seems it would be quite efficient, no? Because you have these loops, and then you're producing simultaneously a whole bunch of these mRNAs that are useful during a fast process, all in one go. You don't have to undergo multiple cycles, keep undergoing meiosis and again and mitosis and whatnot. In one go, from this lamp brush chromosome, you can produce many copies of these transcripts that are important. So it would seem that, the with evolution, these cells figured out a way to have an efficient pipeline to produce a lot of mRNAs that are useful during critical events such as oogenesis. Yeah. So that was about lamp brush chromosomes, and I have some images here that I thought would be interesting for you guys to check out. Um, yeah. So here you can see this is a lamp brush chromosome taken from uh, I think this one is from the frog, and it is of a lower mag, so you can not see it as clearly, and you can see these sort of this green color. This is actually from RNA pol staining. So I mentioned how the loops are highly um, sort of transcriptionally active, right? So this lamp brush chromosome here, it's stained for RNA pol 2, and it's almost marking out the entire structure. So that really shows how transcriptionally active it is, that you can almost map out a structure of histones using RNA pol staining. So that's how active it is. And this is another image, and they have also, this was, they have some other stuff that they're looking at, but they've also marked out nucleoli here as well. Those little dots are nucleoli. And this is interesting. So this is from a Jove protocol where they sort of looked into how to isolate these lamp brush chromosomes. People can do it in different ways, from um, using some microdissection techniques um, to, I think, just some special centrifugation techniques and stuff. So here, this is a sort of zoomed in, or basically a higher mag image of a salamander lamp brush chromosome from the oocyte of a salamander. They used an axolotl salamander, so it's this guy, and I thought I'd show a picture because it's so cute. There is this type of salamander. It's a little different from normal salamanders because it stays in this phase. It does not mature to look like the lizard-like thing that a salamander looks like. So this chromosome, the lamp brush chromosome, they isolated from the oocyte of this kind of an axolotl. And the green is staining RNA pol 2. The blue is staining your DNA. And this is the size of a salamander a lamp brush chromosome, and the frog one actually is a lot smaller. And what's interesting is you can see these loops that they've pointed out, right? So these are actually the loops where the transcriptional activity is super high. So that is about lamp brush chromosomes. Again, if you have any questions, please let me know. Does anyone have any questions? Okay, sounds good. Then I'll move on. So the next type of chromosome, right, the higher order structures that we're talking about right now is a polytene chromosome. So these are found typically in salivary glands of dipterans, or basically insects, and certain insects. And the polytene chromosomes were first reported by Balbiani. You guys have heard of Balbiani loops. So that was Balbiani who discovered them initially. And these chromosomes that are they are an exceptional case of the cell cycle. So what happens in them is that chromosomes in a diploid nuclei, they are typically you have duplication, right, during S phase to form another set of chromatids. But what happens at the end of S phase is that during mitosis, or basically the cell doesn't go into mitosis, what happens is that the DNA strands don't segregate. So after being duplicated, they actually just remain completely paired and parallel to each other. And cell division doesn't occur, so the cell doesn't go into M phase, and they remain in interphase. And in interphase itself, you are able to see this polytene chromosome. And this process of, replication without undergoing mitosis, in this case, it's called endoreplication. Some people also call it endomitosis, and in which this chromosomal DNA will undergo mitotic replication. The strands don't separate, and you have many, many rounds that can give rise to, so it's just like DNA replication, right? But they just get arranged next to each other. So what happens if you arrange enough of them next to each other is they start producing this distinctive banding patterns. And in this example, for instance, if it occurs in larval salivary glands of many species of Diptera, like in Drosophila, salivary glands and stuff, this actually increases the production of mRNA for, in this example, it's a glue protein. It's this mucinous protein. And this is what the larvae use to anchor themselves to the wall during pupation. So it's very convenient for them because suddenly they have so many, many copies of this particular gene. So they can just directly transcribe them all at once and produce a lot of glue protein in one go. So it's convenient you don't have to, duplicate the cells. You don't need many, many rounds of mitosis. Instead, you can just produce more copies from the increased copy number of that gene. So it's convenient. And this diagram here shows the chromocenter where they're sort of attached, and then you have these different chromosomes that have been doubled, and they are all attached here. Regions, there are regions where they're separated, and then there are regions where they are compacted together and they form these banding patterns. So yeah, so aside from increasing the volume of the cell's nuclei, because obviously if you suddenly have so much DNA replication taking place, but the cells are not dividing, it causes an increase in the cell's nuclei. It can also cause an increase in the cell's size. Obviously, as we mentioned, polytene chromosomes or cells that contain polytene chromosomes can have a metabolic advantage because they can have multiple copies of a gene, and this permits a higher level of gene expression. So in Drosophila, for instance, the chromosomes, as I mentioned, have this in the larval salivary glands undergo many rounds of endo-reduplication to produce a lot of this adhesive mucoprotein or this glue protein before pupation. and it helps them stick. And the large size of the chromosome is because of the presence of many strands of, as I mentioned earlier, right, the compacted bands when they come together, they're called chromonemata. So the name polytene, which means many stranded, comes from there. And they are around this size, length, and diameter. And the polytene chromosome can have, now you can see here, dark bands and light bands. So dark bands, and these are called the interbands, and they are darkly stained. Obviously, interbands are lightly stained. That's why they're called that when it comes to nuclear stain. So what would that mean? If something is darkly stained versus something is lightly stained with a nuclear stain that should ideally stain everything homogeneously, what would that mean? That in this region, there is more binding sites, and this region, there is less binding sites. So this is more compacted. So there are less binding sites that are available while or to intercalate to, and here there are um, altogether more binding sites that are accessible for the nuclear stain. So the dark band contains more DNA and less RNA, and the interbands actually contain more RNA and less DNA when it comes to duplication, when it comes to transcription. The bands of polytene chromosomes also can become enlarged at certain times to form swellings called puffs. So this is called puffing. And in the regions of the puffs, the chromonemata actually uncoil and open out to form some loops. So I think there was something here. This is where they separated. So you can actually see I didn't include the image. Okay. So if the region uncoils, what would happen is they form these loops, and these loops actually make the DNA more accessible again. So this is, yeah, and that would indicate, the puff would indicate the site of active genes where mRNA synthesis is actually taking place. So this is just an image of the same thing that I just explained with the bands and the interbands. Yeah. This is another set of images that I found. So this one, this is in some insect which has polytene chromosomes. And so here they've sort of just detected it as an image. This is like a contrasted image with a nuclear stain. Over here you have fluorescent, so the RNA DNA hybrid regions have been stained over here, presumably with antibody. And then this is the DAPI image. In the DAPI image, I have to say theirs is a little overexposed. Naito, you would have been able to see the banding pattern a little more clearly. And then you can see the merged one. And this is the image in phase contrast where you can see the banding. And these asterisks that they've marked out, those are some parts where this fluorescent detection of these hybrids is seen. So they were looking at some micronuclear bodies actually, and they are seeing that these RNA DNA hybrids are sort of enriched in these areas that they have marked out, and you can see them here as well. So, and you can check out this paper if you're interested. Right, okay. So that was all about the higher order structure of chromosomes, specialized chromosomes that are found, lamp brush and polytene. We talked about histones, and we talked about sort of how DNA is bound to histones, the various histone proteins, hats and HDACs, right? Just to recap. And so now, if one were to set out to study these chromosomes, or to study all these different ways in which chromatin is arranged, how DNA protein interactions take place in the form of uh, when transcription is taking place, you have these RNA uh, RNA, DNA, and DNA protein hybrids that show up when you have, say, RNA pol 2 relocated to a region where genes need to be actively transcribed. And so if we want to map out where all you have these protein DNA complexes in gene, um, uh, or where all you have chromosomes bound to certain proteins in the in the genome. How would you do that, right? What is the technique that people have used? So I mentioned a couple of times while I was sort of going through the earlier slides that there's this technique called CHIP or a CHIP assay. So this is chromatin immunoprecipitation. Okay, and I'm sure you guys have heard of it. You know, people say, oh, they did CHIP-seq or they did CHIP-PCR. So what is CHIP actually, right? So you guys know what immunoprecipitation is. It's essentially you bind something with an antibody and uh, you bring it down. So suppose you have a protein in a solution. You can have a bead that's bound to an antibody that binds to that protein, and you bind it and you pull it down, or you precipitate it out. It may not be bound to a bead, maybe you just precipitate out the complex. So that is immunoprecipitation, right? You separate out something from a mixture of stuff to purify it out, to enrich it. That is immunoprecipitation using an antibody, of course. So CHIP does the same thing. It utilizes antibodies that selectively recognize and bind proteins, and this includes histones with modifications, different transcription factors and cofactors, and it gives a lot of information about chromatin states and gene transcription. So a combination, so then once you do this precipitation, a combination of proteomic analysis and different molecular biology techniques are used alongside the main CHIP um, steps. And that gives us the ability to understand gene expression and regulation in cells or different tissues. So typically in CHIP, it's used to identify the relative abundance of a specific protein or a specific protein modification, like say a histone and maybe a modified histone at a certain region in the genome. So CHIP can be used to answer a lot of questions about the interactions of proteins and chromatins. So say histone that is bound in some region of the chromosome, or maybe some SMC proteins that are found in some region of the chromosome, or maybe it is even some transcription factor that is important that localizes to some region in the genome, and we want to know, okay, if I were to say activate the cell, this transcription factor is clearly having an effect, but where is it in the genome? So you can answer that question. You can move, like somehow sometimes we can mutate a gene and look at sort of what's binding to that gene and what mutate its promoter, mutate its enhancer. In this case, you can actually go the other way around. You can find a transcription factor and look for the gene that it's binding to, right? So it's sort of like an opposite method. And so it can be used to answer a lot of scientific questions involving the interaction of proteins and chromatin. Like for instance, it can be used to compare the presence of various proteins at various loci in the genome. Or you can map various proteins across a genomic region of interest, or quantify protein binding to an inducible gene in response to a stimulus. Like I mentioned, say you are working with a T cell and you activate the T cell, then with some some stimulus or the other, and you see that okay, this T cell is now producing this XYZ proteins and it's like secreting them. So you can figure out with the help of CHIP, if that is the question that you're interested in, okay, where is the protein binding, which gene is it binding to, and you know, even when it's binding, how much is it binding. So you can do a lot of different assays with the help of CHIP. So initially, what do? You, if you have a cell, you have to cross-link. So cross-linking is done with the help of formaldehyde, and that will essentially stabilize your structure. And so what formaldehyde does, it's called fixing, right? You fix with the help of formaldehyde or paraformaldehyde, and that preserves the cell in like a lifelike way. So now if you fix a cell, the cell is dead, but it sort of cross links the proteins, DNAs, everything, and it keeps it in a place. So we do this a lot in microscopy as well. If you have a cell and say you give it some treatment, and 5 minutes after the treatment, you want to see what the cell looks like. So imaging it immediately 5 minutes after may not be always very practical if you have other things to consider or multiple, parameters that you want to look at. So what you can do is you can actually just fix the cell. That will keep everything as it is, even if the cell is no longer alive, and it will not let the cell move on with um, any other whatever metabolic or cytoskeletal remodeling, whatever, any sort of change will not happen as the cell will now be dead. And then you can permeabilize it and stain it and so on and so forth. So in this case, what these guys can do for chip is you can cross link it. So you will stabilize the DNA and protein complexes or interactions that were there in these cells, and then you will perform chromatin fragmentation. That can occur with that can be done with um, uh, I think it can be done with mechanical methods, and also it can be used, you can use nucleases. Then you can immunoprecipitate. So suppose there is some protein bound to some region in the gene that you're interested in. You use a protein, so you use an antibody against that protein and immunoprecipitate out that DNA protein complex. And then that DNA, if you're interested in that DNA, then you can purify out that DNA from the complex and perform DNA analysis. So that you can do a qPCR to look at the relative levels, or you can do a ChIP-seq. So essentially, you can perform, next generation sequencing on that DNA to know what is the sequence of DNA that was attached to that protein of interest, right? So, in fact, there are two types of chip. I also did not know this. I learned this when I was preparing these slides. There are two types of chip that can be carried out. So, there is native chip and there is cross-linked chip or X chip. So, based on kind of what you need or what starting material you have, you can use N chip or X chip. I think X chip is much more common as far as I'm aware. So N-chip is where you don't have fixing. So no fixing or native chip. You don't use formaldehyde or any other cross-linking agent. Instead, what you do is you just purify or isolate out the native chromatin from cell nuclei, as you can see here, and you digest it with MNase. MNase is, it's like a nuclease. And that cuts the chromatin in, it cuts the DNA in regions and separates out the fragments for you. And there are regions that are protected from nuclease activity and regions that are not. So that's what helps you digest the chromatin. And then you can whatever, release your supernatant and then use it for your IP studies and so on and so forth. The issue with this one is that sometimes you have to have a lot more starting material to be able to get enough good native chromatin. So I don't know, people may not favor that based on what their starting material is. But if there is for some reason maybe some sensitive component that you cannot cross-link, or um, yeah, then maybe you can do that. Also, another thing with N chip is that maybe you might lose if it's a transient phenomenon. It's something that just comes and goes. You can fix it, but if it's N chip, by the time you actually get to taking out the chromatin, maybe that interaction is gone because the cell was still alive while you were doing this. So by the time you do it, that sort of um, time control is lost. So yeah. X-chip, on the other hand, we use chemical fixatives like formaldehyde to cross-link the protein of interest to the DNA, and then you can fragment. So fragmentation can be done, as I mentioned, in a mechanical method using sonication. So sonication essentially uses really high-frequency sound waves to disrupt the walls of your cells or the membrane of your cells and release materials, or you can also use nuclease digestion. Uh, and the advantage of this is that it can be used with both histone and non-histone proteins, and it, as I mentioned, generally requires less starting material in comparison to N-chip. It also minimizes chromatin protein loss during extraction, and it can allow you to detect a transient protein interaction. So you're not losing your like your control over time that you have to have if your experiment requires it. in an X chip. So this is the X chip protocol. Formaldehyde cross-linking in your cells. Then you can sonicate or do nucleus digestion. Again, you will separate out your supernatant, do your chromatin IP, and then you can use DNA purification and then go for sequencing or qPCR. So I hope that is clear for you guys on how chip works. Um, does anyone have any other questions now, or I can move on? I think now I have a bunch of like MCQs and stuff that we can move through. If you guys are fine. I hope everyone is still there. I can't see. Mm. Okay, all right. So let's start with the questions. Please message the questions or say it out loud by unmuting yourself. Anything is fine, whatever you're comfortable with. So let's get started. Which of the following is not an interaction found between histones and DNA? Do you guys know? I hope you can hear me. Okay, I have one answer. Does anyone else want to try? Come on, guys. Who else is there? There are other people. I hope you guys are awake. Okay, anyhow. So, you're right. It is B, which is covalent bond. So that is not an interaction seen. I mentioned earlier. You have H bonds, nonpolar, dipole interactions, salt bridges, so on. But covalent bonds are not formed between histones and DNA. Okay. Histones with DNA are found in what form? Tetramer, octamer, dimer of two heterodimers, trimer of three homodimers. Okay. I have one answer. Does anyone else want to try? So you're right, it is octamer. You're right. Cool. So as I mentioned, right, you have your H3H4 and then you have your H2H2B and they come together, form a heterodimer, then they form a tetramer because two of them join, and then those tetramers come together to form an octamer with the DNA looped around it. Great. So H1 histone is also known as what kind of histone? There is a very common description of this. Do you guys remember from what I taught and what was in the coursework?
Linkage.
That's correct. Really good. Yeah. So it is known as the linker histone. So why is it the linker histone? Because it helps link the DNA to the nucleosome core complex, right? So to the octamer, it helps the DNA stay bound. So that's why it's a linker. Great. Okay. So next question. Which is true about histone 1? So I just went over this. What do you guys think? Anyone? You can give it a shot. It's okay if you're wrong. Okay. So one answer has come in B. Does anyone have any alternate answer? So I will spill the beans. Actually, the answer here is D. So it's not B. Why? I'll explain. So firstly, let's go through them one by one. So in A, it is a part of the nucleosome bead. So the bead in this case is obviously the octamer. So as I mentioned earlier, H1 is not part of the octamer. It is what is sort of sits on top of the octamer and helps bind the DNA to the octamer. So it attaches two nucleosomes. That is also incorrect because two nucleosomes are spaced apart significantly, and each nucleosome or each bead has its own H1 and they do not bind to each other. It attaches DNA loops to each other, like that's not true. And it attaches DNA to nucleosomes. That is the correct one, okay, because of its linker function, because of the way it interacts with the entry and exit points of DNA on the octamer, okay, on the histone octamer. So the answer here is D. I hope that is clear for everyone now. Okay, so histones have a net dash charge. This one I went over. Typically in homeostasis, okay, people are answering and everyone is correct. It is positive. So good job. Yes, it has an overall positive or plus charge. So that is because of the basic residues like lysine and arginine that are found on them. And that helps it bind to the DNA better. Cool. Next question. So some histones can be modulated via charged modifications, such as acetyl groups, for instance. So some proteins that perform these modifications are, so I talked about them uh, today, and they're also in your coursework, in short. Yep, someone has got it immediately. Hats and edge tags. That's absolutely right. So histone acetyl transferases transfer the acetyl groups, and HTAGs are the deacetylases that remove the acetyl groups, and that can modulate the charge of these histones, which can then modulate how well they are able to bind to the DNA and if the DNA is loose or tightly packed in those regions. Great. Okay. Next question. Which of the following is not a histone? Easy one. What do you guys think? Okay, someone is saying A. Does everyone agree with A or does someone have any other answer? Yeah. So, yeah. So, unfortunately, A is not correct. It is C. So, A is H1. H1 is a histone. It is a histone. It is just not part of that core octamer, right? The actual one that's not a histone that I just invented is H3B. That doesn't exist. So you have H2A and H2B, you have H3 and H4, and you have H1, okay, which is the linker. H3B is not a histone, okay, it doesn't exist. All right, so good. I hope that's clear. All right, we'll move to the next question. What is the diameter of the beads that are found on the beads on a string structure? Do you guys know or remember? So yeah, I hope others also answer, but you guys are doing good. So the answer is 10 nanometers. So one person has said C, which is 30 nanometers. Actually, the diameter of the beads or the nucleosome, that is actually 10 or 11 nanometers. Okay, and those are further compacted, and the next level compacted structure, that is 30 nanometers. But the beads themselves have a 10 or 11 nanometer diameter. Okay. So the answer here is D option, 10 nanometers. Okay. All right. Next one. The dsDNA takes how many turns around the nucleosome core particle? Yeah. So that's right. The answer is C. It's 1.67. This is just like a, sort of knowledge-based question. So that 147 nucleotides translates to about 1.67 turns around the nucleosome core particle. ठीक है. All right, next question. Which is false about histones? Let's see if you guys can get this one. Anyone, any idea? So this one is maybe a little bit tricky. So I'll walk through the options one by one. Yeah. If anyone remembers or has some idea in between, please feel free to answer. Okay. So the first option A. Okay, yeah. Someone has given D option. So you're right. It is D, and I'll come to why it's D. Okay. So, which is false about histone. So, A, they can undergo PTMs or post-translational modification. So, this is completely correct. They can undergo PTMs. B option, can they undergo these modifications or PTMs after binding to DNA? So, that is correct. They, in fact, can undergo these modifications even after they've bound, right? HDACs and HATs themselves are an example of this, of proteins that help the histones undergo such modifications. They have a positively charged surface. This we have covered very well now. And finally, D, which is the incorrect option because they have a flexible end term actually where the PTMs take place. So that's just something that's good to know about the histones. I think in the structures that were shown in the coursework, right, there was how the tails are sort of free flying out of this structure. And there's many of these tails. So in that, the end term is going to be the one which is quite flexible and quite amenable to these different PTMs, either before or after it's bound to DNA. So that's good to know. And that's the correct option. So D is the option here. Okay. All right. 11. Is it true or false? Oh, true or false, I mean. Hats can lead to transcriptional coactivation. Is this true?
It's true.
That's correct. So, does everyone agree? Yeah. So, I hope everyone understands why it's leading to transcriptional coactivation, right? You have an acetyl transfer, you have neutralization of charge, you have loosening of DNA, you have more accessibility of transcription factors. So, hats can lead to transcriptional coactivation. So, another true or false question. Histone variants are found in the centromere. So aside from the traditional histones that we know about, Are there variants that can be found in the centromere?
True.
Yeah, that is correct. Good job. So this was, nicely, I think an example was given in your coursework, and you may have read about it separately as well. But can you guys name one histone variant that's found in budding yeast?
Senpei.
Yep, yep, good job. So you clearly remember that nicely. So SENPA is histone variant that's found in the centromeric region in budding yeast, and it actually helps with um, correct attachment of the microtubules to the centromeric region via the kinetochore. And I think, yeah, I think SENPA is a variant of histone H3. So different histones can have variants. So this one is one of histone H3. So good. I hope you guys all now know this. Okay, next question. So polytene chromosomes are found in which type of cells typically?
Shall I even?
Yeah, that's right. That's good. Yeah. So that's the correct answer. It's B option, insect salivary glands. Great. That was an easy one. All right, what about this one? The characteristic banding pattern of polytene chromosomes is due to,
Option B.
yep. Does anyone have any other answers? Yeah, here my options were a bit weird, but anyhow, that's the correct answer. It is B because you can have these alternating regions of euchromatin and heterochromatin, which forms these bands and interbands. Great. All right, next question. So the lamp brush chromosome, they're typically found in which type of cells?
This time
I didn't quite hear you, but which uh
B.
B, right? Yeah. So that's correct. Yeah, and in the chat also, you're right. It's option B. And I thought maybe you guys might get confused by human germ cells, but I think you guys are all much smarter than that. So oocytes of humans, you wouldn't see it because it's specifically not found in mammals. So yeah, great. All right, question 16. So the extended loops of the lamp brush chromosome that you saw in those fluorescence images as well, it's associated with.
Option A.
Anyone has any other answers? I think everyone agrees, huh? So it's right. Absolutely, it's higher levels of transcriptional activity. Like you guys, right, remember about how you have higher copies of, say, um, the mucin gene, right? The sort of gluey gene. So that helps with producing more glue protein. So you can have higher levels of transcriptional activity in those regions. Cool. What is the primary function of hats? So again, this will be easy because we went over this quite a lot.
Option B.
Anyone else? Great. So yeah, you're right. It is option B. It acetylates lysine residues on histones. Great. And how does histone acetylation by the hats affect chromatin structure? What do you guys think? Anyone? So, uh, wait, hold on. Has anyone messaged the answer? No. Okay, so maybe this one I'll just re-explain because so that it's clear for all you guys, so you can answer this question easily if it were to come up in some test or something. So as it was seen, in the previous question, the primary function of HATs is to acetylate lysine residues on histone. So that's option B in 17. In 18, we are asking, how does that affect the chromatin structure? So HATs are transferring a negatively charged acetyl residue onto the histones, that's neutralizing their positive charge. So what is happening then? Histones are no longer as much net positive as before. So if they're less positive, they are going to be less attracted to the negatively charged DNA phosphodiester backbone. So they're going to be less tightly packed together. That's going to loosen the chromatin naturally, right? And that will promote gene transcription. So the option is B. Histone acetylation by HATs loosens chromatin and promotes gene transcription. Okay, so that is the answer. All right. Right. Question 19. Which of the following is likely a consequence of HDAC inhibition? So think of what we just learned about HATs, right? The mechanism that I outlined for you guys. So HDAC inhibition will lead to what? So what do HDACs do? And if I were to inhibit an HDAC, then what would happen?
Option A.
Hmm. So maybe try again. Think about what the HDAC does typically, and if you were to inhibit it, then what would happen?
C.
That's right, yeah. So the correct answer here is C, and I'll just quickly explain it so that it is clear for everyone. So HDACs remove the acetyl groups. So what is happening there? That you are increasing the net positivity of the histones, allowing them to interact more with the negatively charged DNA. That is causing them to be closer together. They're compacting, and it's less um, available or less accessible to transcription factors. So transcriptional repression or gene silencing takes place if you have a lot of HDACs or a lot of HDAC activity. Now, if I were to inhibit it, then the opposite of that would happen. Basically, it would be like a hat. So it would increase, if I were to inhibit a HDAC, you would have increased histone acetylation and gene activation. Okay, so I hope that is clear and this mechanism is clear to everybody. You just remember it about just remember like the charge, how it happens. If you remember that an acetyl group is negative, then everything else falls into place because you know that histones are positive and acetyl is negative and DNA is negative. So then you can just do that plus minus and figure out, okay, this is what is happening with HATs, HDACs, and their corresponding inhibitors. Okay, so I hope that's clear. I'll move to the next question. What is the primary purpose of a CHIP assay or a chromatin immunoprecipitation assay?
C.
Someone is saying C. Okay. Any other answers?
Option B.
Yeah, so that's actually the correct one. It's option B. I'll just so as I showed the chip assay sort of slides, and that you would have seen that uh, we are using an antibody against something, right? So an antibody typically will be against a protein. So in this case, that protein is some protein that's bound to chromatin. Okay, so it could be a transcription factor, it could be a histone, it could be some modified form of a histone. Okay, and what we do here is that you want to map out where these protein DNA interactions are taking place. And you can do that with the help of an antibody to immunoprecipitate out the protein DNA complex. And why in vivo? Because essentially this interaction is taking place within the nucleus of a cell that is alive, that or in a tissue uh, from some organism, whatever your experiment is accordingly. And that is the native state of them. And to that, you're adding formaldehyde, which fixes them in a lifelike uh, way. And then you're taking out these proteins, complexed proteins with DNA and studying what DNA is there. So it is identifying the protein DNA interactions in vivo. Okay. So that is the primary purpose of chip. Now, I think there are probably 100 different variants that people are doing, but this is the overarching goal of it. Okay. All right. So in a chip assay, what is the role of formaldehyde or other cross-linking agents? So I mentioned this like just now. Anyone? So maybe you guys are a little tired. So this one is option C. Okay. It is to stabilize the protein DNA interactions before immunoprecipitation. So essentially, you are fixing or cross-linking um, with the help of formaldehyde. Um, so I don't know what percentage formaldehyde is typically used in this chip assay because I don't perform it most of the time in my work. But when it comes to cells and stuff, you typically don't go more than like 4%. So 2 to 4% is more than enough to fix cells for flow cytometry or imaging assays. So I use around 3% in my assays to fix cells. So you will use some, you know, amount of diluted formaldehyde, which will help you fix your cells and that will stabilize your protein DNA interactions. So that will fix them. That will not allow any further interactions to take place. Whatever was binding at say T equal to 2 is going to stay bound. Okay. And then you can look at it. So that is that. 21 ka answer is C. All right. Chromomeres are best described as,
Option A.
anyone else? Any other answers? I think everyone agrees with you. You're right. It's option A, which is they're highly condensed bead-like structures visible on chromatin, and they're often corresponding to regions of gene regulation. Okay. So good. I think that is all the questions I have for this part, and I think I have just a couple of little things, little questions for the part about SMC. So SMC was covered in so much detail. I didn't really want to go into too much because, you guys already know it all from the extensive course material. So I thought I'd just give a little recap and then we can do like some 5, 6 questions on it and then end for today. Okay. So as you guys must remember from your coursework that the SMC or structural maintenance of chromosomes are these non-histone proteins and that are highly conserved and they're ATPases that have some fundamental roles in higher order chromosome organization and dynamics in organisms all the way from bacteria to humans. Okay, so they're highly conserved and they have they're energy dependent, their ATPase function. So SMC dimers, they adopt a two-armed structure. So like this, they have a two-armed structure with a central hinge domain that connects the long coiled arms. So the central hinge domain is here, and this connects with the long coiled arms, and these are the ATPase head domain. So the ATPA binding head domain is at the distal end. So, oops, okay. And the head domain of SMC protein, it's structurally related to this ABC or this ATP binding cassette or ABC protein. So the domain of the ABC transporters, that's what it is similar to. So this is your coiled coil region, or this one you can see, coiled coil region, hinge region, and these are your head domains that are attached to each other. And there is another complex that helps attach them as well, called the glycine subunit. So let me come to that. So ATP binding and hydrolysis can actually modulate the engagement and disengagement of the head domains. So these guys, from attaching to one another, ATP binding can modulate them attaching to um, each other in that region. Um, and they have an important role in regulating the dynamic interactions between SMC proteins and DNA. Okay. Condensin 1, okay, so condensin, cohesin, these are all SMC family proteins. Condensin 1 has the capacity to introduce positive superhelical tension into DNA with the help of ATP hydrolysis, and maybe it's also giving some positive gyres or loops. And a single DNA molecule assay actually revealed that there's a dynamic and reversible compaction of DNA1 that's supported by condensin 1. So this is condensin 1 over here. So this is the one that's important for binding and looping DNA to condense it. So this you will see a lot of condensin activity um, during prophase, where you have condensation of chromosomes. Then cohesin. Cohesin initially was found to hold sister chromatids together by essentially holding the two DNA duplexes close together in this ring-like structure. And it has composed of SMC arms, and it has this non-SMC glycine subunit. So over here, it has a non-SMC glycine subunit that helps it hold these regions together. And the SMC proteins might actually use a lot of diverse different interactions, either intramolecular within molecules of this protein itself, or intermolecular protein-protein interactions to help tether, fold, and manipulate the genome in an ATP-dependent manner. So just like histones, SMC proteins are also very important for organizing the genome into chromosomes, for organizing regions specifically such that there are some regions that are more or less accessible in an ATP dependent manner, which is different from histones. And these are some electron micrographs of these different SMC proteins that you can see here. Yeah. Right. So these are the questions. So I think we can just jump directly into the questions. If anyone has any questions about SMCs, do let me know. I can tell you what little I myself know about them. Um, yeah, but I think I've covered the basics. Yeah, cool. I think one thing I forgot to mention is that the glycine subunit is also quite amenable to these post-translational modifications. Yeah, that is something that can also then modulate the head region, which can then therefore modulate how well it binds to or doesn't bind to DNA. Yeah, cool. So, all right. First question. My apologies, I don't have the question numbers here for some reason. What is the primary role of SMC proteins in chromatin organization?
Option B.
Yeah, good. So that's correct. It's option B, facilitating chromatin cohesion. So cohesins can do that, and chromosome condensation. The condensins can do that. So that's great. That's option B. All right, next question. What is the significance of sister chromatid cohesion during mitosis? It's kind of a take back to previous lectures. Anyone? Okay, someone says A. Hmm. Hmm, not entirely. So, ideally, nothing should prevent chromosome segregation because if you prevented chromosome segregation, then you would have aneuploidy, right? So, definitely, you don't want to prevent chromosome segregation. You want to make sure that it happens accurately. So, here the correct answer is B. Okay. So, if you have proper cohesion of sister chromatids, if they're attached to each other correctly, then later when they are separated from one another, and the protein complexes are all in the right place, the enzymes involved will be able to separate them correctly and attach them correctly to the spindle fibers or the microtubules, and that would ensure accurate chromosome segregation. So the answer here is B. Okay. All right. Next question. In SMC proteins, the ATP binding head domain is formed by. So I showed this earlier. I think there are like
Option
different
C.
names. Yeah, yeah, correct. So there are different names, I think, for the motifs based on the kind of complex that you're dealing with. But in this example, it was Walker A and Walker B motifs uh, that were the ATP binding head domains. So great, good. Um, Good to see that you guys are paying attention. All right, which is the next one? So which of the following proteins bridges the head domain of the two SMC proteins in cohesion complexes?
Option C.
Yep. So you're right. It is option C. It's glycine, which can be MCD1, SEC1, or RAD21 in terms of yeast. Okay, this one is a little throwback. Cohesin is cleaved at the metaphase to anaphase transition, right? So who does that? Does anyone remember? No? So cohesin helps with attaching these sister chromatids together, right? So at some point, when we are separating the sister chromatids from one another, we would want to make sure that cohesin is cleaved, right? And that is going to be at the metaphase to anaphase transition because at anaphase is where you pull the sister chromatids to opposite ends of the cell. So separase is the enzyme that cleaves cohesin. So that is good for you guys to know because it's very important in remembering when it comes to the checkpoint transitions and it comes under what is this thing called? The cell cycle control with respect to checkpoints. This particular reaction is quite important because if separase does not cleave properly and cohesin is not cleaved properly or at the right place or enough, or for all of the chromosomes, then you're not going to have proper separation. Okay, second last question, I think. What happens to sister chromatids after the cleavage of cohesin is done by separase? This is easy.
Option C.
Yeah, great. So that's exactly right. They move apart towards opposite poles. So I hope this sort of, flow of things is clear now. Okay, last question. So there was one more complex, right, which is a little bit less known about, which is the SMC5-6 complex. So it's known for its role in which of these following functions? And you guys remember? There were cohesins, there were condensins, and now there's SMC5-6. Someone says D. D, I don't think D is right. I don't think that's a function of SMC5 or 6. I'm pretty sure it's C. Yeah, yeah, it is C. That's correct. So, C is the correct answer here. It is involved in DNA double strand break repair. Um, I think it participates in homologous recombination, that mode of double strand break repair. It also, I think, promotes replication fork stability, and yeah, it's important in meiosis and telomeres, maintaining telomeres close together. So clustering of telomeres, SMC5-6 is involved in that. So yeah, option C is the correct one over here. Okay. So I hope that's clear. I hope that jogged your memory and understanding of what you've gone through in your coursework. Yeah, that is it. Um, so, okay. I hope that is clear now for everyone. And I hope, yeah, I hope it was useful for you guys. And I'll see you guys next time on Monday. And yeah, it'll be an interesting lecture next time because it's, I think, about DNA damage and dysregulation and cancer. And since that's some of the things I work on, I'll be I'll put in some more interesting information there. So I hope you guys all join next time. Yeah. All right. So okay. Thanks everyone for joining, and I'll see you guys next class.